Thank you very much for the introduction and the opportunity to give this talk here. I hope I don't keep you too long from your coffee break, which is starting in 3 minutes. So I'm going to talk about the genetic component, the germline genetic component of drug sensitivity in cancer. And starting with what is cancer? I mean, cancer is a genetic disease. Oh, why is it actually?
So you can, I had that problem earlier. You can reset it.
Can you just, sorry.
So what we did is
I just tried it before and it was working.
Yeah, so we do this. By the way, everybody's going to stay through the coffee break to see this, right? We do this. And then you do this. And then you go back to this. And now it should work.
Oh, perfect. Thank you very much. I'm really sorry. So basically, cancer is a genetic disease which is driven by copy number variation, fusion genes, and mutations. And actually, those differences are called somatic mutations. So the difference between healthy and cancer tissue, and those are accumulating during the lifetime. And interestingly, those could be used as drug sensitivity markers, so which is the basis of personalized medicine and targeted therapies. And how does germline now play into this equation? So germline are inherited differences, so they're potentially identically in every healthy cells, with a few exceptions like B cells. And those are mostly considered as toxicity markers because they would affect the healthy tissue as much as the cancerous one. But that is not the full story. The full story is actually that you look at also the somatic mutations within a germline background. And that, interestingly, becomes again a marker of drug sensitivity and not only toxicity. So for this analysis, we used the GDSC screening, which is a competitor project of CCLE, the Cancer Cell Line Encyclopedia, the European version basically. They screened 1,001 cell lines, 265 drugs, and also did an extensive molecular characterization of mutations, combination, methylation, gene expression. And their objective is to identify somatic markers of drug sensitivity. Looking at our phenotype, we're looking at a concentration and cell viability on the y-axis, and usually this is looking like a sigmoid curve. And our phenotype is the area above the curve. So a large area above the curve would correspond to a cell line which is very sensitive. So the sigmoid curve would be shifted to the left-hand side if you have cells which are strongly responding. So for example, the blue one would be a sensitive one, the purple cell line would be a semi-responder, and the red one would be a non-responder in our observed concentration range. And the question is, what's the difference between those three cell lines? What is the genetic marker which is causing this difference? And as I said before, the equation is somatic mutation plus germline plus the interaction. And looking at the somatic mutation, we just separate patients by the small star, so by differences we have in the tumors. So the germline component is we would sort them by their genetic background. And this would be like green people versus black people. Oh, sorry. The last component is the drug, the interaction. So basically, you would look what is a green with a star. So basically, the star within a population, within the green people, and would that be more sensitive or resistant? And this would be again a marker of drug sensitivity. So looking at the somatic component alone, here we built a predictive model. We were predicting 1 minus AUC versus observed 1 minus AUC and calculated Pearson correlation. And we just see here like the drugs, how they perform, the 265 drugs. Some of them are performing quite well. And for some others, we do a bit overfitting. And there's also some where we don't capture much. And if we add the germline genetic component, you get a lot of those spikes and they go up. So you get an improvement in performance. And I would like to talk about this big spike here, which is 17 AAG, where we get a lot of improvement when we include the germline component. And looking at both here, with the Manhattan plot, in green you see the somatic component, in gray and in black you see the germline genetic component, and you have here a large spike, which is NKO1. So NK1, what is that? It's an enzyme which is metabolizing actually 17-AAG into active state, which then becomes a very potent HSP90 inhibitor, which is a housekeeping gene which is important for folding, helping folding various client proteins. It's a chaperone. If you don't have functional NK1, you have also slightly inhibition of HSP90, but not as potent. And looking at that TT, that is the broken allele, you have less sensitivity in TT, but CC and CT allele are quite good in responding. And what is here, if we look at the NK1 expression, there's no separation with them. But very interestingly, we see again an interaction between those. So this is not only an additive effect, actually you even get slightly response in the broken allele if you consider the NK1 expression. And here you see this again, expression and response, and AUC is going up. It's not just a flat line. So this is only the working allele. We're looking at where we have working NK1, and we compare it with modern HSP90 inhibitors, which are directly targeting HSP90. And here you have a flat response, and interestingly, you have here a crossing over. So if you have high enough NK1 expression, actually this might be a good choice, a good therapeutical choice. And also very interesting, NK1 is differentially expressed in cancer versus normal. So this means cancer relies on it independently of that it's also metabolizing this drug, which then becomes very potent. So now I was showing you an example of germline, and we were basically finding an interesting interaction with expression. But here now I want to look at interactions between germline and somatic. And therefore I would like to jump into one example. Again, the NAP kinase pathway, RAS, RAF, MEK, ERK, which ultimately leads to cell proliferation, and afatinib, which is inhibiting this RTK EGFR. And we were looking at one somatic mutation, RSG1, RAS suppressor unit 1, and you don't see any difference between mutant and wild type. And there's also nothing reported with RSG1 inhibiting RAS. So looking at the germline component now, this is MUC19. There's a small difference, but there's still nothing to see actually. I mean, that would be like really imagine things. But if we look into basically RSG1 within the germline background AAA, you get a massive response, and that's very different. Bottleneck is you have only a few points which are making this box plot, so you subset your already sparse set, and that is the statistical problem you have with interaction, and we are just beginning to actually be able to do such experiments. Now, looking at, I mean, we found more interactions actually with the volcano plot showing the resistant and sensitive ones and p-values at different cutoffs, FDR cutoffs. And afatinib is here our example with RSG1 and with MUC19 germline association interaction. And we find also gefetinib, which is another EGFR inhibitor, which is below the 30% FDR threshold, but at least pointing in the same direction, giving us some confidence that there might be actually something true down to it. And those are 35 interactions, and we filter by EQTL and missense mutations, to reduce the number of tests, because otherwise comparing all germline versus somatic, that would be quite extensive. To summarize, I would like to highlight germline alone is a marker of toxicity, and it's studied quite well in that area. Somatic mutation is also studied as a drug sensitivity marker, but the interaction of germline and somatic is again a sensitivity marker, and that is not well studied, particularly because we didn't have the sample size to do those studies, and hopefully in the future there will be more data available that we actually can do this. And we're just beginning to start to do those studies. And here I showed you one interaction which we found by accident. We were looking into a germline strong hit, and actually found an interaction with the germline with the gene expression of NK1. And also the interaction which I showed you was afatinib with MUC19, which is mucin, and which is a secreted protein which is like causing a protection layer around tumors, and RSG1, which was not reported in any way with afatinib. And last but not least, I would like to acknowledge quite a lot of people, particularly Francesco Paolo Cansale, who equally contributed to this work, my supervisor, Julio Saez Rodriguez, who's also in the room, and Oliver Stegel, who contributed with his knowledge about Limix and GWAS and basically his pipeline to do those kind of studies. So thank you very much for your listening and staying and keeping away from coffee. And I would happy to take questions.
We will be breaking for coffee upstairs. The next talk is at 10:45.